By now, you should have non-Mendelian genetics written at the top of the next blank page as your title. So as we move through this, I would like you to write in your notebook what is highlighted on the slides. Also write and draw out the problem sets and the Punnett squares. And this will include additional information such as phenotypic ratios. Today, we're going to be looking at non-Mendelian genetics. So many characteristics have more complex inheritance patterns than those studied by Mendel. They are complicated by factors such as codominance, incomplete dominance, multiple alleles, and environmental influences. Today, we are going to look at examples of incomplete dominance and codominance. So the first one we're going to look at is incomplete dominance. So make this your section title. During incomplete dominance, what you have is that neither allele is completely dominant. Thus, the word incomplete dominance. So you end up with a heterozygote that shows an intermediate phenotype or a blend of the traits. So let me show you an example. You have snapdragons. Red snapdragons would have two red alleles, and white snapdragons would have two white alleles. Now, if those were crossed, you would get not one of these dominant, rather a blend of the traits, resulting in a pink snapdragon. So go ahead and write your definition of incomplete dominant. Okay, I'm going to go ahead and move on. If at any point in the presentation you feel that you need more time, just raise your hand and I will pause the video. Here are some extra examples of incomplete dominance. So here we have a green betta fish and a blue betta fish. And when crossed together, they create a teal betta fish. So this is an example of incomplete dominance. And again, we have our example with the snapdragons. Okay, so we're going to practice a Punnett square using incomplete dominance. So unlike Mendelian genetics, dominance is incomplete. Okay, so when we are talking about this problem, the color red is going to be represented by a capital R, and we are using carnations for our cross. So the color red of carnations is incompletely dominant over or with white carnations. So the hybrid or heterozygote is a blend resulting in the color pink. So I'm going to give you a second to write that down. Okay, so I want you to notice that both alleles are capital letters. So for a red carnation, both alleles would have to be red. So you would have a capital R and a capital R. For a carnation to be white, it would need two white alleles, which are going to be represented by a capital W. So it would be capital W, capital W. So the reason we're doing this is because neither of these are dominant over the other. Okay, so let's make our cross. We are going to be crossing a red carnation with a white carnation. So for your box, you can just make the 4 squares, or you can make the extra squares as I prefer to do to keep things in line. Okay, so now that you have your box, at the top corner, you're going to write a capital R that represents a red carnation. And since it is red, it has two red alleles. So you're going to also put capital R for the second allele, which is for a red carnation. Now along this side over here, we're crossing it with a white carnation. So white carnation must have two white alleles. So your parents over here are capital W for white carnation and another capital W for a white carnation. So you are going to follow the same rules you would for a monohybrid cross regarding how you bring the alleles down and over. So for our first box, we get R, W. Go ahead and note that down. And since R and W are the heterozygote, and neither is dominant over the other, this would result in a pink carnation. So we're going to do the same for the rest of the boxes. So R and W for this one. So again, you will get a pink carnation since you have two alleles and they are incomplete dominant alleles. And finally, for these two, you are going to bring them over and across. So you are going to get R and W, again getting a pink carnation since it has two alleles that exhibit incomplete dominance. And finally, the last one, R, W. Again, a pink carnation. So if we look at this, how many out of 4 of the offspring are going to be pink? 1, 2, 3, 4. So all of them, 4 out of 4. So our phenotypic ratio, the physical characteristic, is going to be 100% pink. Make sure to get that written down. Okay, so we're going to move on to codominance. So this is your section title, so go ahead and write it. So keep in mind that the prefix co means with. So the phenotypes for both dominant alleles are co-expressed in the heterozygote. What that means is that both dominant alleles traits appear in the heterozygote. Go ahead and get that written down. So some examples of this are cattle coat color as well as feather colors in chicken. So with cattle, you can have a pure red or a pure white coat. However, if you breed these or cross these together, because they display codominance, their offspring would display both of the traits. This coat color is called roan. So notice that both dominant traits are displayed here. So co meaning with. So one dominant trait is being displayed with the other trait. Another example we can look at is in chickens. So black chickens must have 2 alleles for black feathers, and white chickens must have 2 alleles for white feathers. However, if you were to cross the two, what you will end up getting is what we call, or what is called, a blue chicken. And the reason it looks blue is because it's speckled. So the speckling of the white and black dominant colors showing through in the fur make it appear blue. So both the dominant black and dominant white colors are being displayed in the heterozygous offspring. Now we're going to do a Punnett square example with codominance. Example. In some chickens, black feathers, capital B, are codominant with white feathers, capital W. If a chicken has one allele for black feathers and one allele for white feathers, rather than like Mendelian genetics where it would display dominant, Both white and black feathers are codominant, so both white and black will show through. So it will be a speckled chicken, displaying both traits, kind of spotted black and white. So up here you can see the chicken with black feathers, chicken with white feathers, and here is your spotted chicken. Okay, so let's do an example between two heterozygous parents. So keep notice, with codominance, you're still using all capital letters because one is not dominant over the other. So we're going to put our parents on the top and the side. So first parent has one allele for black feathers with a capital B. So go ahead and make sure you draw your Punnett square. And it has a second allele for white feathers. Goes capital Y, put that in your Punnett square. Now down the side, we're going to put parent 2, so it has one allele for black feathers. We're going to use capital B. And its second allele is for white feathers, so we're going to use a capital W. Okay, so finally we are going to use the same method. We're just going to pull down and pull over. So the first box, you are going to get B, B. So this has two alleles for black feathers, so this offspring will have black feathers. In the next box, you pull it over and down, and you get B and W. This has one allele for black and one allele for white, so it will be speckled, both black and white. Now we're going to go to the bottom row. So this chicken has one allele for black feathers and one for white feathers. So it'll be a speckled chicken also, displaying both white and black feathers. Finally, we're going to go pull from over and down. And the last one has two alleles for white feathers, meaning that it will be a chicken with white feathers. So now we're going to look at the phenotypic ratio. We have one black chicken, two speckled chickens, and one white feathered chicken. So our phenotypic ratio would be 1 to 2 to 1. So 1 to 2 to 1, with 25% being black, 50% that would be speckled, 25% with white feathers, making it a ratio of 1, 2 to 1. So now that you have your notes for non-Mendelian genetics regarding incomplete dominance and codominance, I want you to complete your practice problem sets for both of these types using your notes if you need to. So keep in mind, incomplete dominance, neither is dominant over the other, so they end up blending. And with codominance, co meaning with, both dominant alleles both appear within the organism.